In today's webinar, we'll focus on novel cell-based assays to enable immunotherapy drug development for checkpoint receptors. The field of immune checkpoint-mediated immunotherapy was founded by these two gentlemen, James Allison and Tasuku Honjo, who were awarded the Nobel Prize in Medicine in 2018 for their pioneering research on the T-cell inhibitory receptors CTLA-4 and PD-1. And in this work, they demonstrated that inhibition of these checkpoint pathways allows T cells to eradicate cancer cells. So effectively, that inhibiting these particular pathways removes the brakes that inhibit T cell activation and therefore allow us to modulate the immune response to destroy cancer cells. So this particular modality has been utilized very successfully for the clinical development of therapeutics targeting PD-1 and CTLA-4, resulting in the therapeutic antibodies Keytruda and Opdivo that we all are familiar with. But in addition to CTLA-4 and PD-1, a number of additional inhibitory receptors have been identified, and many groups are looking for approaches to inhibit these as additional therapeutics using the same approach with blocking antibodies. Now, the challenge with developing therapeutics to these receptors is that it's important to understand the specificity of the molecules on the particular receptors of interest. So assays are needed to allow us to look at those receptors in a very specific way. So our goal as a company has been to develop such tools, and I'll give an example of an assay that we have designed focusing on initially PD-1 and a signaling assay that allows us to look at inhibition specifically of PD-1. So in this particular cartoon here on the left, what we're showing is that PD-1, as we all know, is expressed on the surface of T cells. And when it engages its ligand presented on the surface of the tumor cell or the antigen-presenting cell, this results in an intracellular event that phosphorylates the C-terminal tail of PD-1. And this particular event results in recruitment of the SHP2 phosphatase, which itself results in dephosphorylation of the T-cell receptor that is mediated by proximal signaling kinases. So this particular event that occurs here at this ITIM and I-switch domains is an early event that that is physiologically relevant and is something that we can measure by looking at recruitment of the SHP to the C-terminal tail of PD-1. So we've done this using our EFC technology, which is shown in this cartoon here at the bottom. Essentially what we have is a fragmented reporter that's split into two pieces. The small enzyme donor is about a 40 amino acid peptide, whereas the remainder of the reporter we call enzyme acceptor. And the way that we use this system is that we fuse the two proteins of interest to these inactive fragments. And when the interaction of the two proteins of interest brings these two fragments into close enough proximity, they'll reconstitute an active enzyme. And that enzyme can then be detected by addition of substrate. So the way that we designed our PD-1 assay is we developed it as a co-culture model where our PD-1 is expressed as a full-length receptor on the surface of Jurkat cells, and it's fused to this small enzyme donor tag. In the same cells, we're co-expressing the SHP phosphatase fused to the enzyme acceptor. So in the absence of any ligand, we have very low signal in the assay when they're co-expressed, but when you co-culture these PD-1 expressing cells with ligand presenting cells, then you get a gain of signal, which can be detected by addition of your substrate. Similarly, if we block this interaction with antibodies or other molecules that inhibit either the receptor or the ligand, And we're able to actually see a reduction in signal in a dose-dependent manner. So our PD-1 signaling assay is available in two formats. It can be used using cells in continuous culture, or we have an assay-ready format, which we provide using frozen ready-to-use cells. And so the protocol for that is shown in this particular slide, where for an anti-PD-1 therapeutic, we would thaw the PD-1 cells and seed them into the assay plate and allow them to recover overnight. We prepare our antibody dilutions and add them to the assay plate for an hour. Then we thaw and add directly our PD-L1 expressing cells to the assay plate and co-incubate those with the PD-1 expressing cells for an hour. And then we add our detection reagent to the assay plate and allow that to incubate for 3 hours before reading the assay on a plate reader. The important thing here is that this is all homogeneous, so there are no transfers to other plates, no spinning, none of those types of things. It's a very simple assay. So what you can see is that the assay is highly specific. In this diagram, what we're showing is the response to PD-L2 ligand presenting cells, and you see a nice dose-dependent increase as you increase the number of cells. Where by contrast, if we take a cell line that's expressing a ligand for a different checkpoint receptor, in this case the HVEM ligand, we see no response in the assay. Similarly, if we look at specificity from an antagonist perspective, we see that we get a nice dose-dependent response with our anti-PD-1 antibody, but an antibody to an unrelated receptor, in this case TIM3, produces very little activity in the assay. Finally, what we've shown is that these assays are suitable for detection of not only biologics, like our anti-PD-1 antibody shown here, but we're able to see inhibition with small molecules that inhibit the PD-1, PD-L1 axis, such as this low molecular weight inhibitor. And by contrast, molecules that are not specific to PD-1, such as the small molecule negative control, which is specific for EGFR, we see no inhibition in the assay. So this is an example of our PD-1 signaling assay. We developed assays for a number of other checkpoint receptors as well. They use kind of the similar signaling modality. So in addition to the inhibitory receptors, I think we're all aware that T cells also have a variety of co-stimulatory receptors that provide a second signal to activate T cells. And these receptors fall into two different classes. The first is the immunoglobulin superfamily, and these are very similar in structure to the PD-1 receptor. These are typified by CD28 and ICOS in this particular figure. And they also signal through a similar modality to PD-1, where they recruit an SH2 domain containing protein, which I'll show you in a moment. The remaining of the receptors, costimulatory receptors that have been identified to this point, all fall in the TNFR superfamily, so the TNF receptor superfamily, and these predominantly signal through the NF-kappa B pathway. In all of these cases, for the costimulatory receptors, from an immunotherapy perspective, the goal here is to develop agonist antibodies that allow us to push the gas pedal, if you will, on T cell activation to further stimulate the immune system to destroy cancer cells. So if we focus on our CD28 and ICOS costimulatory receptors, which I showed you in the previous cartoon, and I mentioned that these do signal through SH2 domain containing proteins similar to our PD-1 signaling assay. So in the case of our JERCAT CD28 signaling assay, shown on the left, CD28 signals through GRAB2 recruitment, whereas the ICOS assay on the right signals through the P85 subunit of the PI3 kinase. In both cases, what you can see is that these assays are robust and enable rank ordering of agonist antibodies. And you can see the differences in both efficacy and potency between these different commercial antibodies. So in the case of one of our TNFR superfamily receptor assays, this is an assay that targets OX40. We have the option of developing assays that target either the canonical NF-kappa B pathway, which is shown here in this cartoon on the right, which signals through I-kappa B and the P50 RelA complex, or we can use the non-canonical pathway, which signals through Nick. And in the case of OX40, it does signal through both pathways, so we elected to use Nick accumulation as a measure of pathway activation. And the reason for this is that the Nick protein, which is the NF-kappa B inducing kinase, is present at very low levels when the pathway is inactive. But when it becomes activated, you see an accumulation of Nick in a dose-dependent manner. And so what we've done is we've engineered these cells to express Nick fused to our enzyme donor. And so it's a very simple assay to engineer. And we have also engineered overexpression of the OX40 receptor. And in this case, what we can see is that we have a nice dose-dependent gain of signal of OX40, or in this case, Nick accumulation in response to soluble OX40 ligand. And this response occurs within about a 5-hour incubation time. So for many of the co-stimulatory receptors, it's been documented that clustering of the receptors actually improves the efficacy of agonist antibodies in vivo. And this clustering event occurs primarily through Fc gamma receptors. Now, Fc gamma receptors are a family of endogenous receptors that are expressed primarily on immune cells, and they're known to bind to the Fc portion of antibodies. And there are a variety of different Fc gamma receptors that are expressed on different immune cells and can engage different antibodies. But for the TNF receptor superfamily, it's been documented for a number of these receptors that engagement of the inhibitory Fc gamma R2b receptor, which is expressed on specific immune cells, is critical for the efficacy of agonist antibodies for these targets. And what's thought to happen in vivo is that this activation of downstream signaling pathways requires receptor oligomerization, and that by cross-linking of the IgG-Fc region to these Fc gamma R2b receptors, it results in multimerization, especially with more than 2 antibody molecules. And this multimerization appears to facilitate clustering of enough of the TNFR receptors to activate signaling. So in order to evaluate the effect of Fc gamma receptors on the activity of agonist antibodies, we have engineered a number of Fc gamma R expressing cell lines that can be used in co-culture models to test these agonist antibodies. So as an example, we have used our OX40 signaling assay, which has a molecule, pogalizumab, which has been demonstrated in vivo to require cross-linking. So this is the right-hand end of the cartoon here. So as I mentioned, pogalizumab is an OX40 agonist antibody that in vivo is not active in the absence of Fc gamma R2 engagement. So in our assay, what we see is that the soluble antibody is effectively inactive. If you treat pogalizumab with a secondary antibody that cross-links the Fc portion of the antibody and then add that to the cells, you do see some activity in the assay, which is better than the soluble. But the most efficacious way to present the antibody to the cells was co-incubation of the cells with our Fc gamma R2b expressing cells. So you see a nice dose-dependent and enhanced potency and efficacy when using the Fc gamma R2b cells in co-culture with our OX40 signaling assay. Importantly, clustering can also promote activation of our co-stimulatory receptors from the IGSF family as well. In this case, I'm showing an example with ICOS. But what's interesting is that it appears to be an antibody-dependent phenomenon. This is true for the TNFR superfamily as well, that not all agonist antibodies require clustering for potent responses. In part, this depends on where the actual epitope is that the antibody recognizes on the receptor. And this is demonstrated here in this particular slide, where we can see on the left, we're testing a commercial anti-ICOS antibody in the assay, where the soluble antibody shown in pink is not particularly potent. But when we co-incubate the cells with, in this case, a mouse Fc gamma R2b presenting cells, because this is not a humanized antibody, we see a nice shift in potency and an improvement in efficacy as well. Whereas with this second commercial antibody, we don't see as robust or as significant a difference when we co-incubate with the Fc gamma R2b cells. We already get quite a robust response with the soluble antibody. And it appears that the two antibodies do recognize different epitopes on the receptor. So up to this point, we've really talked about assays that support development of therapeutics targeting the adaptive immunity branch of the immune system. As we've talked about checkpoint inhibitors and checkpoint agonists, we have assays that allow one to evaluate those particular therapeutic modalities. But it's becoming increasingly appreciated that the innate immunity branch, which has a variety of other immune cells that are important in the tumor microenvironment, is another area for ripe for modulation. So many people are focusing on toll receptor agonists and STING agonists, but another area of growing interest is blocking this particular axis, the SIRP alpha CD47 axis. Now this SIRP alpha CD47 axis is being called a macrophage immune checkpoint, and SIRP alpha itself is a receptor, an inhibitory receptor expressed on the surface of macrophages and dendritic cells that effectively is involved in phagocytosis of foreign objects. So CD47, which is the ligand for SIRP alpha, is expressed on nearly all cells, and it serves as a don't eat me signal on these cells, and by doing so it represses signaling via SIRP alpha and prevents phagocytosis. However, what's been observed in many tumor types is that CD47 is upregulated, and this allows the tumors to escape phagocytosis. And this upregulation occurs in many tumor types, especially in hematological malignancies such as AML and myelodysplastic syndrome. Now, a number of groups have determined preclinically that blocking this axis with anti-CD47 antibodies or SIRP alpha FC basically promotes tumor killing, and a number of groups are now showing this in early clinical data as well, that this promotes phagocytosis in the tumor. Another benefit of anti-CD47 blockade has been that it enhances adaptive immunity as well by priming an anti-tumor cytotoxic T cell response. So very similar to what we've talked about for the immunoglobulin superfamily receptors, the SIRP alpha receptor also has this modular domain in the C-terminus that responds to phosphorylation upon ligand engagement, and that phosphorylation site recruits SHP phosphatases. And this phosphorylation and SHP recruitment event leads to inhibition of myosin accumulation at the phagocytic synapse, and this is what prevents phagocytosis. So this is the molecular MOA that we chose to develop an assay for the SIRP alpha CD47 signaling axis. And again, we used a similar approach that we did for our PD-1 assay using our co-culture SHP recruitment model, I won't belabor this since the concept is the same as it was for PD-1 and the other assays, except that in this case we're using a Jurkat cell that's presenting CD47 as the ligand. So many groups, when they've talked to us about developing assays for SIRP alpha CD47, are interested in also evaluating it using plate-bound CD47 Fc. So what I'm showing here is an agonist response with the plate-bound CD47 Fc. And you see that we do get quite a robust assay window, about a 13-fold window. The EC50 is around 4.5 micrograms per mil, so quite a lot of protein is required. Whereas with the cell-presented ligand, we get a signal to background of about 22-fold. And again, in the antagonist mode, we see a signal to background around 8, but we get a much more robust assay window. And with the same anti-CD47 antibody, also a much more efficacious and potent response using the cell-presented ligand. So this is the modality that we prefer for this particular signaling assay. So we have developed this assay using a stable line that expresses SIRP alpha very stably over 45 passages. You see that the expression by flow varies by less than 20%, and we see a very stable functional response over that 45 passages as well, where the IC50 and the signal to background vary by less than 20% RSD. Similar to our PD-1 assay, we see that we have excellent assay specificity. Again, here our anti-CD47 produces a very robust inhibitory response, whereas antibodies targeting unrelated checkpoint ligands have no activity in the assay. And finally, because there is a lot of interest in this particular target in the CMC space, we wanted to show that the assay is also able to detect altered forms of the antibody, such as this heat-treated forced degradation sample. And you see that we can detect potency differences when the molecule is perturbed in this manner. So I'll just conclude with the benefits of the PathHunter signaling assays that I've shared to this point. They are simple and biologically relevant homogeneous assays. They provide a rapid response with good assay windows. And they're highly specific and sensitive assays suitable for discovery through lot release. They are compatible with biologics as well as small molecules. And they're suitable for screening both agonists and antagonists even for the assay formats that I've shown you. While the focus is on immunotherapy, they can also be used for other therapeutic areas as well. They're also, the protocols are extremely easy to transfer, and many of the assays are available in our ready-to-use format, which is the frozen ready-to-use, allowing much easier transfer to our CRO partners. So I wanted to focus on one additional assay format that can be relevant for immunotherapy discovery, and that is cell-based assays for bispecific antibody engagement. And the concept is very simple, as shown in this cartoon. What we're doing is expressing the antigens that the two arms of the bispecific antibody recognize as fusions to either the enzyme acceptor or the enzyme donor, which in this case is called PK. And they're expressed on the surface of the same cell, such that when the bispecific antibody is co-incubated with these cells, the arms of the bispecific bring the two fragments, the two receptors together, and then bring the EA and PK into close enough proximity to reconstitute a functional enzyme. So this represents a physiologically relevant context of presenting the two receptors. So in some ways, it's more physiologically relevant than an SPR assay. And we found our clients have really found these are useful to actually allow them to rank order molecules and rapidly evaluate the effect of different linkers or chemistries in a single readout and compare that data to their SPR data. And because the assay is a homogeneous assay, it's amenable to high throughput, so it's suitable for early discovery, but its simple homogeneous format also makes it amenable for a QC lot release. So in this particular example, we're showing some typical data for this type of assay in this graph on the left. So the dark blue and green curves correspond to two different bispecific antibodies for this particular dimer pair, if you will, which is anonymized here. And what you can see is both of those antibodies are active in the assay. However, monospecific antibodies that recognize the two different arms of the bispecific and either combined or treated separately are unable to activate the assay. So the assay is highly specific only for the bispecifics themselves. So up to date, we have generated over 10 different receptor combinations involving immune checkpoint targets, such as PD-1 and LAG3, PD-1 TIM3, etc. And we've also developed some murine analogs of the same dimer pairs. I mentioned that this is a highly specific response, and we've also applied this same approach to development or monitoring of antibodies that mediate dual inhibition of oncogenic receptors, such as the c-Met EGFR bispecific antibody. And this particular combination, the work that we did in collaboration with this group, it was actually published, and you can certainly read the paper and learn more about these assays through this publication. So finally, if you don't, these assays are all published on our website, but if you're not able to find the combination that you're interested in, new assays can be engineered through our custom assay development program. So I'll close by highlighting some of the assays that I've talked about. These are also shown on our website. These on the left are the examples of our functional cell-based assays from either the immunoglobulin superfamily or the TNFR superfamily. I mentioned the clustering cell lines that can be used for evaluating agonist antibodies. And then, of course, some of the assays that we have on our website for our binding for bispecifics. So I would encourage you to reach out to us and in any questions that you have, or if you're in need of custom solutions, we're happy to help you there. Thank you so much for your attention, and we hope to hear from you soon.